The immune system. Angelina Jordan, RN, BSN. This in-service is to provide dialysis nurses a better understanding of the immune system. The learning objectives are to understand the types of immunity, to understand the cells of the immune system and their roles, to understand the immune responses, and to understand the phases of the immune response. Immunity. In natural immunity, factors are present prior to exposure to an infectious agent, and all respond nonspecifically. Factors include physical barriers, phagocytic cells, and natural killer cells. Specific acquired immunity. Immune responses occur only after exposure to a foreign substance. Foreign substances, called antigens, induce specific responses. The objective of the immune response is to destroy them. Specific immune responses are possible because certain cells of the immune system possess receptors that can recognize individual antigens. Cell-mediated immunity. Immune responses in which targets are attacked directly by the immune system cells. Humoral immunity. Immune responses that are mediated by antibodies. Cells of the immune system and their roles. B cells make antibodies and mediate humoral immunity. T cells lyse target cells. Helper T cells promote proliferation and differentiation of B cells and CTLs. They also initiate delayed type hypersensitivity. Macrophages promote proliferation and differentiation of helper T cells and CTLs by serving as antigen-presenting cells. They participate in delayed type hypersensitivity. They phagocytize cells tagged with antibodies and phagocytize cells in the effector stage of delayed type hypersensitivity. Dendritic cells promote proliferation of cytolytic T cells and helper cells by serving as antigen-presenting cells. Basophils mediate immediate hypersensitivity reactions. Neutrophils phagocytize foreign particles, especially those tagged with IgG. Eosinophils attack helminths and foreign bodies that have been coated with IgE. They contribute to immediate hypersensitivity reactions. Immune responses. There are 5 features. Specificity. Cell-mediated and humoral immune responses are triggered by specific antigens, and their purpose is to destroy the antigen that triggered the response. Diversity. Our immune systems can respond to millions of different antigenic determinants. This is possible because our immune systems have millions of clones of B and T lymphocytes. Memory. Exposure to an antigen affects the immune system such that re-exposure produces a faster, larger, and more prolonged response than did the initial exposure. Time limitation. Immune responses don't last indefinitely. They are time limited. In the absence of a continuing stimulus to generate more active B cells and T cells, the immune response fades. Selectivity of antigens. Our immune systems normally target only foreign antigens, leaving potentially antigenic molecules on our own cells untouched. Sparing of itself is possible because as T cells develop in the thymus, cells that can react with antigens of self-origin are eliminated. Phases of immune response. Specific immune responses can be viewed as having 3 main phases: recognition, activation, and effector. Recognition. The recognition phase occurs when a mature lymphocyte encounters its matching antigen. Specific immune responses begin with antigen recognition by B cells and T cells. Activation. Antigen recognition causes the lymphocyte involved to become activated. The activated lymphocyte then undergoes proliferation and differentiation. Some of the daughter cells differentiate into cells that actively participate in the immune response, attacking the source of the antigen. Other daughter cells differentiate into memory cells, thereby preparing the host for a more intense, rapid, and prolonged response in the event of antigen reexposure. Effector. In this stage, the immune system attempts to eliminate the specific antigen that initiated the response. With cell-mediated or antibody-mediated immunity, several effector mechanisms can be involved. References: